Namaste friends, welcome you all for the biology session. This is Manjunath JS from CET Sankalp course. Let us continue with the chapter Digestion Absorption part 3. In the previous class, we discussed about pancreas, stomach and intestinal glands. Let us continue with the gastric glands. Right, as we already discussed about the role of the gastric juice, right, where exactly the different types of cells which produces different types of cells in the stomach which produces what is that example we can take that parietal cells produces HCl. So that is the main role of the gastric glands which secrete HCl, and along with that the enzymes. So enzymes will be produced by mainly the peptic cells. What is that? Peptic cells which produces enzymes. So as we all know that those enzymes like we can say pepsinogen and renin, which will be in inactive form, and they will be converted into active form in the presence of HCl. So they gets activated activated in the presence of HCl. So this in the previous class we discussed. Let us take the what is exactly the content of the gastric juice. So gastric juice, which mainly contains HCl, pepsin, renin and mucus. There are certain neck cells which produces mucus, right? So these are the things which will be there in the gastric juice which is produced by gastric gland. Next, intestinal glands. And these glands mainly play a role, right, in the production of intestinal juice. Intestinal juice, which is also called as, very important to note that, succus entericus. Intestinal juice which is also called as succus entericus. So this generally mainly with juice with water, mucus, salts, and along with that digestive enzymes. Let us take that within a while. Here the enzyme, which is a non-digestive enzyme produced by intestinal gland, already we have mentioned that enterokinase, which is also called as enteropeptidase. Enteropeptidase, which activates all the proenzymes, that is inactive enzymes produced by pancreas. Activates proenzymes which are produced by pancreas. So next, so what is the role of enterokinase? Which is a non-digestive enzyme produced by intestinal glands that presents in the succus entericus, which activates the proenzymes of pancreas. So next, this intestinal gland which also secretes the enzymes, right? The enzymes in the intestinal juice are aminopeptidase. These are very important thing, you should keep it in mind. Dipeptidase, aminopeptidase, sucrase, maltase, lactase, isomaltase, lactase, isomaltase. So these are the enzymes which mainly they act on carbohydrates. Acts on carbohydrates, and the production of intestinal lipase. Intestinal lipase, right? So which is produced generally acts on lipids, acts on lipids. And along with that, here the one important thing I mentioned, that in the pancreas, that is you should keep it in mind, right, that which will going to produce a pancreatic lipase, which is a strongest lipase. Pancreatic lipase, which is a strongest lipase, that is steaphase, right? So this, along with that, it also includes nucleotidases, which acts on nucleotides, and nucleosides. So these are the enzymes which acts on what is that, nucleic acids. Generally we'll say derivatives of nucleic acids. Nucleotidase acts on nucleotides, right? And nucleosidases, right, which acts on nucleosidase, which acts on nucleosides. So we'll take that in the further physiology of digestion. So these are the enzymes present seen in the what is that, intestinal juice. So ಇಷ್ಟು enzymeಗಳು what is that, they play a major role in the digestion of substrates, right? Next, in this, the important thing you may get this question is that, right, the proenzyme, mainly the trypsinogen, what is that, gets activated into trypsin in the presence of what is that, in the presence of enterokinase. So this is the one important question we can expect, that is enterokinase, which is also called as enteropeptidase. Let us move on to the next. The concept is histology of alimentary canal. Right. So these are all the glands which play a major role, right, generally in what is it, the secretion of digestive enzymes, right, that which helps in digestion. So if you're going to what is it, the observe that throughout the alimentary canal, the wall layer, right, there are what is it, the four basic wall layers, right? There may be what is it, the structurally slightly variation, but these four basic wall layers are common. So what are those? The outermost, what is it, the layer which is called as serosa, right? Right, so whenever we're going to what is it, the consider this the outermost layer, each of these layer will be with certain type of tissue which play a major role in digestion. Each of these what is it, the wall layers, which will be made up of what is that, certain type of what is it, the tissues. So those tissues play a major role in digestion. Right, so we can say that all together all these what is it, wall layers, which will going to one or the other way perform what is it, or play a role in digestion. Serosa is the outermost layer, which is mainly what is it, the connected with mesentery, right? Outermost layer connected with mesentery, that is what is it, the body wall connected with mesentery. And this is mainly consisting of single layer of squamous epithelium. So here you should keep it in mind that whenever this the organ, we're going to say that the visceral organ, right, visceral organs, in those visceral organs we can say that, right, that is the generally mesothelium, epithelium of the visceral organ, which is called as mesothelium. And next layer, so here some of the connective tissue, with some connective tissue will be there. Squamous epithelium, which is called as mesothelium, and right, the connective tissue serosa is consisting of connective tissue. And inner muscularis, that is next to that, the second layer, which is called as muscularis. So second layer, that is the muscularis, which has a two, what is it, the wall layers, or I can say that they made up of two layers, muscle fibers, two layer of, what is that, muscle fibers. What are those two layer of muscle fibers? Inner circular, right? Generally the inner layer, which is made up of, that is the circular layer, right? And the outer, if you're going to consider that, that is made up of, what is that, longitudinal layers. How exactly we can imagine this? You can draw like this completely. Just I will take that in the straight line, right? And these are the outer, what is that, longitudinal layers. And internally, in which we can see, what is that, the circular layers on this, right? So like that, these layers which will be there arranged, and these layers play a major role in, what is it, the creating the alternative contraction and relaxation. One important note here, should keep it in mind, in almost all the, what is it, the organs, the muscularis is with two layers, outer, what is it, the circular, outer longitudinal, inner circular, outer longitudinal, inner circular. So in stomach, it varies. Muscularis is made up of three layers. Note, muscularis is made up of three layers. What is that? That is the additional layer is a oblique layer. So we can say like this, that is outer longitudinal, what is that, the middle circular and inner oblique muscle, and third one, which is the inner oblique muscle, right, inner oblique muscle layer. So these, what is that, the layers which will be present seen in, what is that, the stomach. Note, right, we may expect this question. So next, so in this, the third layer, what we're going to see is submucosa, right? Submucosa, which is mainly made up of loose connective tissue. Submucosa made up of loose connective tissue, and there will be nerves, what is it, the supply along with the blood vessels and lymphatic vessels, right? That will be supply of blood vessels and then lymphatic vessels and nerves which will be supplied with this. And in some region of submucosa, right, let me take, what is it, the, here I'm going to erase this serosa layer, right, so that I can write, right, the third layer, that is the submucosa, which will be there in some part of the, what is it, the alimentary canal, we can say, what is that, some with certain glands. In some region, we can say some organs, right, we can see that the glands. Example, in the duodenum, right, in the duodenum, in the submucosa, we can see glands called as Brunner's gland, right. Next, and the innermost layer, which is called as, what is that, mucosa, outer serosa Next to that is muscularis, and then the third layer is submucosa, and the fourth layer is mucosa. So mucosa which lines generally the lumen, mucosa which lines the lumen, where it is mainly made up of what is that, the different types of cells in different organs, like villi in what is it, the intestine, like villi in intestine, and what is it, the rugae in stomach, rugae in stomach, right? As we all know that what is the role of villi, villi which what is it, the generally in the small intestine, in the small intestine, which is what is it, the more the villi are present, finger like projections which are lined by what is it, microvilli, microscopic what is it, these cells which are called as microvilli. So let me what is it, the give that little detail in that. Let me give what is it, the what exactly the microvilli and the brush border appearance, right? See, these cells which will be there generally will take up that in the structure that is here. You can see that these cells which are there, generally the epithelial lining of the villi, if you're going to write like this, just some few cells you can write on which we can see like this. So this, if this is a finger like projection, and that which will be lined by what is that, the microscopic what is that, the cells, these are called as microvilli. So microvilli which gives a brush border appearance, brush border appearance, right? So this is because of the microscopic projections called microvilli, microscopic projections called as microvilli, which lines the villi. So all together this structure, if you consider this is what is it, the villi. So these are the finger like projections which are called as villi. So villi are lined by microvilli, and microvilli which gives the brush border appearance in the small intestine. So what is the role of that? That they're going to increase the absorptive surface area, increases the absorptive surface area. Villi and microvilli increases the absorptive surface area, absorptive surface area. So these cells which also consisting of what is it, the mucosal epithelium also consisting of goblet cells, and these goblet cells along with the glands, these goblet cells which will going to produce mucus, they secrete some mucus which helps in lubrication. Generally we can see that in the esophagus, right? Mainly in the esophagus, the goblet cells, right? The epithelium, mucosal epithelium will be with goblet cells. These goblet cells will going to produce mucus. Next, so these what is it, these cells which will be there, right? Generally what is it, the what we can say that the villi, at the base of the villi in the intestine we can see this region which is called as crypts. Familiar irtira ನೀವು, crypts of Leberkan. What is that? Generally they are called as what is that, the crypts of Leberkan, crypts of Leberkan, right? Crypts of Leberkan, right? Intestinal glands which are also called as crypts of Leberkan. Intestinal juice which is also called as succus entericus. Just you keep it in mind, this is very very important. In the small intestine, what is it, the mucosal epithelium which will be mainly consisting of what is it, villi and microvilli, and same like in the stomach. If you're going to take that stomach, actually in the stomach, these the mucosal lining which are with irregular folds, not like finger like projections, they will be with irregular folds, right? Like this, right? not with a particular pattern, but whereas in the small intestine we can see what is that like this. So these are the finger like projections, right, which is towards the lumen, that is what we can say villi. So these villi are lined by microscopic, what is it, the cells or projections called as microvilli. Whereas in the stomach, the lumen, right, there are the irregular folds. These irregular folds in the stomach, irregular folds in the stomach, that is, or I can say that the mucosal epithelium, which is called as rugae, that is in stomach. So we can expect this question. So mucosal epithelium, right, mucosal epithelium in stomach, which is called as rugae because of the irregular folds. So in this we can see gastric glands. We can see gastric glands. In these folds, irregular folds, we can see gastric glands. So generally this structure, which what is it, whenever we're going to say, right, so which will be like this. So one will be completely longer one will be smaller one. So here, which the so the gastric glands will be there. So these gastric glands, as we all know, that they produce a gastric juice. Different type of what is it, gastric glands which will be there, right? So different types of cells in the gastric glands. And here intestinal glands. So just keep it in mind, mucosal epithelium with the glands. And there are different layers in the mucosa, right? So just I am going to mention, that is, one is muscularis mucosae, muscularis mucosae or mucosa, muscularis mucosae, that is the layer which, what is it, the thin layer, right, and next to that, that is from outside to inside towards the lumen I'm mentioning, that is epithelium, that is mucosa, which has a 3 layers. Muscularis mucosae from outside to inside. Muscularis mucosae, lamina propria, and then that is the last one, mucosal epithelium. So in this mucosal epithelium we can see those glands, intestinal glands, gastric glands, right? So those glands which will be there play a major role in the digestion. So with this we can conclude, that is the small intestine, right, mucosa, where it shows villi and associated structures. So we'll take up what are the structures. So villi will be, what is it, the mainly consisting of lacteals, lymphatic vessels of the villi. Lymphatic vessels of the villi are called as lacteals, which helps in the absorption of, what is it, fatty acids, So let us move on to the next, what is it, the topic, that is the physiology of digestion. So before going to take up this physiology of digestion, we know that generally the enzymes which are involved in the digestion are the hydrolytic enzymes, right? Hydrolytic enzymes. They belongs to the different, what is it, the classes. So one, there are mainly 4 different types of enzymes. What is that? The first one we can say carbohydrases, which acts on or helps in hydrolysis of carbohydrates, and then proteases, which acts on protein, which hydrolyzes the proteins, and lipases, which acts on lipids, or you can say which hydrolyzes the lipids. And the last one, nucleases, what is that, which hydrolyzes the nucleic acids. So these are the, what is it, the 4 different, what is it, the types of enzymes which involved in digestion. So let us take there's some question before going to take physiology, some question related to the digestive tract, that is wall layer. Lining of intestinal wall from outside to inside, from outside to inside. So we are all familiar with that. That is outermost, which is called as serosa, right? And then next to that is, what is that, muscularis, and in that muscularis we can see outer longitudinal, outer longitudinal, right? You can imagine that some pillars which are placed in this circular pattern, right? So just I'm going to give like this, just the lines, these are the pillars, right? And then next to that there will be, what is that, circular layer. So pillar ಆದಮೇಲೆ circular, what is it, the chambers ಇದೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. So that is what actually the outer longitudinal, inner circular, what is it, the muscle fibers will be there in the muscularis. And next that opens here, we can see circular muscle, no, that is the outer longitudinal. Here they have not mentioned, that is the serosa. We can say the first layer is serosa, and next to that is a longitudinal and then circular. So where exactly we can see that longitudinal and circular muscle. And next to that is submucosa, right? Submucosa, that, so next to that layer is mucosa, next to that layer is mucosa. So mucosal layer, which will be generally with epithelium, right? Mucosal epithelium. So answer for that, so they can also give here serosa, right? Serosa, next to that longitudinal muscles, next to that is circular muscle, submucosa, mucosa. So we can expect this question like these questions, right? Next diagram given below represents that is what the sectional view of, what is the epithelium. That is, we can say that the mainly villi, right? Villi, along with that mainly this layer, which is a mucosal layer. Sectional view of the, what is that, the mucosa, innermost layer of the small intestine. And these are, what is it, the arteries and veins. So these are, what is it, the arteries, and here will be the veins, and these will going to form a capillaries. So whenever, what is it, we will going to say this, that is the structures A, which is finger like projection, right? That is a cross section they have taken. These finger like projections are called as what, villi. A is villi, so that we can say here A is villi, in this it is not there. And the C option we can have villi and D. So let us take from, what is it, the A and C. So villi, and then after that internally these structures. So B, which is labeled, that is to show lacteals, lymphatic vessels of the villi. B is lacteal, and C, these arteries and veins, if this is the lacteal, and the complete the lining, if you consider this as the finger like projections, the arteries will take some different color. So just here they will going to form a network, right? So network of capillaries will be there. So this network of capillaries, where it will going to exchange, right? Where it will going to exchange the, or you can say diffusion of food, which going to takes place by means of active transport or by means of passive transport. So this structure which will be there, this is lacteal, right? So B represents a lacteal, and C represents, what is that, capillary network, capillaries. And D represents, I've already mentioned that in this region, what are those, they are present intestinal glands, which are called as crypts, crypts of liver. So answer for that, A is villi, B is a lacteal, C is capillary, D is crypts, crypts which are also called as crypts of Lieberkühn. That is, the answer is A. Next we'll take up physiology of digestion. So just the major points we can recollect here in the physiology of digestion, that is the enzymes are involved. What happens in the, the stomach, that is first from the buccal cavity we can start, right? So let us take from the buccal cavity. As we all know that the digestion of carbohydrates, where it starts generally in the buccal cavity, right? So first I will take in the buccal cavity. In the buccal cavity, or we can also say that mouth, right, where the digestion of or breakdown of starch initiates. The first, it's begin, right, digestion of starch begins in the mouth, right? What exactly the required thing? The starch, we know that the salivary amylase, which will going to produce, right, so generally which is produced by the salivary gland, that acts, right, at pH 6.8, slightly acidic, converts the starch into maltose, isomaltose and dextrin. Isomaltose and dextrin. So generally these are disaccharides. So what is the role of salivary amylase, which acts on polysaccharide and converts that into disaccharides? So next, after that, when food will keep on moving, right, from what is it, the mouth to the pharynx to the esophagus. In pharynx and esophagus, no digestion takes place. Pharynx and esophagus, no digestion takes place. Keep it in mind, because no digestive, what is it, the glands, that there are no digestive glands, hence no digestion will going to take place. No digestion because of the absence of digestive glands. Next, and in the stomach, as we all know that the stomach, where this is very very important, in stomach no amylase, no salivary amylase, or you can say that no amylase, hence no carbohydrate digestion. There won't be carbohydrate digestion. But we know that parietal cells and peptic cells, which mainly play a role in the digestion of protein. Right here, the digestion of protein starts. Digestion of protein starts. Digestion of protein that starts. So this is very very important. With the help of gastric glands, right, the gastric juice, which will be having the proteases, they are going to act on the proteins. And next, here the carbohydrates won't get digested, and then it will going to keep on moving into the next part, that is in the duodenum. This is very very important, right, where carbohydrate digestion takes place. Starts in, what is it, the buccal cavity, starts in buccal cavity, and it won't, what is it, the digest in the stomach. It won't digest in the stomach, and directly it will going to move into the, what is it, duodenum. Duodenum, where that, what is it, the starch, the remaining partially degraded food, that is along with 30%, here we can say approximately some 30% of starch will be digested in the mouth. Remaining, which will be moved, what is it, the into the duodenum, where that starch, with the help of many, what is it, the enzyme. The first is that the pancreatic amylase. This is, you have to keep it in mind that that is secreted by pancreas, which converts the same thing, converts maltose and then isomaltose and dextrin. Isomaltose and dextrin. So now here many other, what are the disaccharides in the presence of enzyme converted into products. Maltose in the duodenum in the presence of enzyme called as maltase. We mentioned no, the enzyme present in the intestinal juice, that is that maltose, which converts maltose into 2 units of, what is it, the glucose, right, that is a monomer of, what is it, the starch. or you can say that polysaccharide, right? So 70% of the starch which will be get digested in the duodenum. So here these are very important thing. Maltose in the presence of maltase enzyme, sucrose in the presence of sucrase, make a note of it, sucrase which is also called as invertase, sucrose which is also called as invertase, that which converts, right, that sucrose acted upon by sucrase converts the sucrose into glucose and fructose, glucose and fructose, right? They may ask which is the enzyme which converts the sucrose into glucose and fructose, sucrase or invertase. Same like that, name the enzyme which produces only what is it, the glucose molecules, only glucose monomers, that is maltase. Maltase is the one which produces what is it, only glucose, whereas the sucrase which is going to produce glucose and fructose, right? And along with that, next we can say that the lactose, right? Lactose, the enzyme which acts on this is a lactase which converts glucose and galactose, glucose and galactose. So glucose and galactose which will be produced by the what is it, the substrate called lactose. And isomaltose, we can also say that this is also which will be converted into what is it, the two glucose molecules, that is isomaltase. Isomaltase is the enzyme which converts, right? And next, the most common monosaccharide, that is what which will be glucose. Most common monosaccharide and simplest monosaccharide is what is that, the glucose. So here next we can take that the digestion of protein. So digestion of protein, that is a carbohydrate, very important thing, where it completes generally almost all, and after that some of the pancreatic amylase will going to act on that. After that intestinal juice which will be with enzyme, they will going to act on remaining or partially digested starch, partially digested starch. So that is what these things will going to takes place. So next, protein, digestion of protein, right? First, buccal cavity, it won't what is it, that takes place because no what is it, the proteases, whereas here in stomach, this is very very important, stomach, the digestion of protein starts, digestion of protein starts. So this digestion of protein which initiated by, as we all know, that chief cells, that is through the production of what is it, peptic enzymes. We can say that the peptic cells which produces peptic enzymes which acts on protein, pepsinogen and prorenin. So these will going to act on what is it, the proteins. What is that, the proteins, they will be activated in the presence of HCl, and the protein in the presence of what is it, the pepsin, after acted upon by the HCl, the pH in the stomach which will be what is it, the 1.8, highly acidic. So in that condition, that will be converted into proteoses and peptones. So protein which will be converted into proteoses and peptones in the presence of pepsin, and the one more, renin we already discussed one, that is it acts on milk protein called casein, right? And the next, in the duodenum, what happens, right? So next, this food which will be what is it, they move on to, that is when the food which is mixed with this gastric juice and what is it, the partially digested food, that is we can say that the semi solid food, right, when completely mixed, that we're going to call it as chyme in the stomach. In the stomach, the food which is present, right, present along with the gastric juice and all other, what is it, the substances of the gastric juice, right, that is semi-solid food, which will be called it as gastric juice. Whereas in the buccal cavity, which will be called it as, right, in the buccal cavity, the food which will be called it as bolus, bolus, along with what is it, saliva and salivary amylase, the food which will be called it as bolus, bolus in the buccal cavity, and the chyme in the stomach, chyme in the stomach. So let me mention here one more word, that is chyle, chyle in the, what is it, small intestine, chyle in the small intestine. Let us take this, what is it, the point later. So now in the duodenum, what happens after, right, when in the stomach, this will be get, proteins will be get converted into what, that is proteoses and peptones. So these proteoses and peptones, which will be further, what is the digested, duodenum receives the proenzymes, that is all the proenzymes of the pancreas, and those proenzymes which will be converted into active form, right, active form, proenzymes of the, proenzymes of, what is it, the pancreas, which will be converted into active form. We know that in the presence of enterokinase. So what happens, first thing is that peptones, actually all these things will going to take place simultaneously. It is not like that first pancreatic, what is it, the amylase, and then later after the other, the enzymes all together, which will going to simultaneously, which takes place, right. Peptones in the presence of, what is it, the carboxypeptidase, that is here we can also write, that is a trypsin. First let me take that trypsin, right. So trypsin, which is the active form, that is a active form of trypsinogen. So this, what is it, the protein in the presence of trypsin, same thing, that will be converted into proteoses and peptones, right, that acts on, and whereas on peptones and proteoses, on peptones and proteoses is very very important, carboxypeptidase, we are all familiar with this, right, carboxypeptidase, right, that is what, procarboxypeptidase, which will be converted into carboxypeptidase in the presence of trypsin. Same like that, the one more enzyme, what is that, that is aminopeptidase, right, aminopeptidase. So these are the activated form, right. So now they are going to act on peptones and proteoses, convert that into amino acids, amino acids, or you can say that dipeptides directly. Sometimes they are going to get converted into, what is it, amino acids, dipeptides, and on this dipeptides, dipeptides, right, definitely we can expect many questions on this. Dipeptides in the presence of dipeptidase, in the presence of, what is that, dipeptidase, dipeptidase, which will be converted into amino acids finally, right. That is the very important thing, where it will going to, what is it, the breakdown, mainly it, what is it, the breakdown, the peptide bond, what is it, peptide bond, which releases, what is it, the, if there are two, what is it, the peptides, what is it, the amino acids which are attached by peptide bond, if that will be released, amino acid, that will be, what is it, the breakdown, and which will going to, what is it, the release the amino acids, that is monomers of proteins, monomers of proteins. Whereas carbohydrates, where glycosidic bond will be, what is it, breakdown by carbohydrases, carbohydrases. So with this we can conclude a protein digestion, protein digestion. Whereas a fat digestion, we are all familiar with that, bile juice play a major role in the fat digestion. So let us take that before going to take that the fat digestion, let us take some questions related to these things. Digestion of protein begins with digestion of protein begins in, as we all know that chief cells are present in what is the stomach, and also required acidic nature will be there in the stomach is 1.8 pH. Hence protein digestion begins in the stomach, right? We have these three, so we eliminated this. Next, in the stomach. Next, digestion of polysaccharide, that is what? A starch. In the presence of amylase, starch gets digested in the presence of what is that? The amylase. We know that generally, right? Maltose, isomaltose, and dextrin. So we are all familiar with this. Really, in mouth, the digestion of what is it? The starch, that is a polysaccharide, begins. So answer, that is a stomach and mouth, right, which will be there in the C option. So 34th question, answer is C. Next question, that is we discussed about this. Study the given flow chart, right, which shows fate of carbohydrates. Means different enzymes will going to act on that what is it, the carbohydrate, and we're going to what is it, they convert that into products. So as we all know that during digestion, the human alimentary canal, right, where this carbohydrates will keep on moving, these enzymes what is it will going to act. What are those? Let us take starch, which will be converted into maltose in the presence of what is it, the generally we'll say amylase. We can also say that, right, salivary amylase. This is very, very important. What is that? Amylase, right, which is also called as in what is it, the mouth, salivary amylase. In stomach, there is no amylase. In what is it, the duodenum or in the intestine, there will be what is it, the pancreatic amylase. Pancreatic amylase in duodenum. So that starch will be converted into maltose, and remaining these things which will be converted starch, lactose, right, the milk sugar, and then sucrose, that is cane sugar, which will be converted. So lactose in the presence of what is that? Lactase. In the presence of lactase. So B we can say that is lactase, and C maltase. What is that? Maltase. C, that is maltase. And sucrose, which can also see all these will be converted into what is the two monomers, which will going to produce what is it, the two monomers. That is maltose always produces two glucose monomers. Lactose, galactose and glucose. Lactose, when it is digested with the help of lactase, produces galactose and glucose. Sucrose produces fructose and glucose. So what is that? Sucrase. Sucrase, the other name we have to search, that is which is also called as invertase. Sucrase, which is also called as invertase. In particular sucrase invertase. ಈಗ ಆನ್ಸರ್ ನೋಡಿ, A is amylase in all the option we have, and then B is what is it, lactase. That is we can see that in the D option, that is lactase. And next C is maltase, and the D is invertase. D is invertase. So this we can say that is D is the answer for 35th question. So once again you just go through that with the flow chart, then you can recollect that. Next, we know that renin is the enzyme, whereas this is involved in the digestion, right? Whereas renin, it is produced by the kidney, right? Kidney, which produces mainly what is that, juxtaglomerular cells, right? Juxtaglomerular cells will going to produce, right? In excretion chapter we can study about this juxtaglomerular cells, JG cells. So JG cells which produces renin, that helps in what is that, mainly activating, or I can say stimulates the adrenal cortex to produce aldosterone, sterone, which play a major role in the reabsorption of sodium ion. So that is maintain the blood volume, or you can say blood pressure. So you can say here, see, Renin, that is actually that acts as a hormone but acts on protein. That is, renin acts on protein but not this one. Wrong. Trypsin, we know that that acts on, what is that, the proteins, which won't acts on, it won't acts on, what is that, starch. Right. Invertase, which is also called as sucrase, acts on sucrose. Amylase acts on lactose. No, amylase acts on starch. Starch. So answer for that is C. Answer for that is C. Let us take next question. Which of the following, what is that, is not the function of large intestine? We'll take that during, what is it, the absorption, but we'll solve this question. So which of the following is not the function of large intestine? Generally, function of large intestine mainly, what is that, some electrolytes along with water, right, which will be absorbed. Some electrolytes along with water which will be absorbed, but no nutrients will be absorbed in the large intestine. So absorption of water takes place and then it produces the mucus where it can lubricate the feces. It stores that temporarily, and then it will be, what is it, the that waste, digested waste, which will going to move into the rectum where it will be temporarily stored, and finally that will be eliminated through, what is it, the anus, right, which will pass through anal canal. So through the anal canal it reaches the anus and that will be eliminated out. So here the nutrient absorption, right, no nutrients will be absorbed. That means digested food like what we can say glucose, right, and then amino acids won't be able to absorb. And here compared to the small intestine, in the large intestine we can see very, what is it, the less, what is it, the scattered villi. Villi will be very, what is it, the less. So finger like projections which will be very less, hence no, what is it, the digestion, sorry, no absorption can be seen. Nutrients won't absorb. So answer for that is B. So 37 answer is B. Food mixes, next question, food mixes thoroughly with acidic gastric juice. What is the meaning of acidic gastric juice? Food plus, what is that, acidic gastric juice, we can say chyme. Correct previous. So that is acidic gastric juice of the stomach by churning movement. That is what one type of, what is it, the movement which will be there in the, just I will mention here, swallowing with the help of, what is it, the buccal cavity. That is in the buccal cavity, generally with the help of pharynx, swallowing with the help of pharynx, and then chewing, right, or mastication which will be there in the, what is that, mouth, right, and then churning will be there in the stomach. That which helps to mix the food. And after that, segmentation will be there in the small intestine. Largely will be there in the small intestine, right. And what is it, the one more we can say that the propulsion which includes actually propulsion or movement which includes peristaltic movement. What is it? Peristalsis or peristaltic movement. So peristaltic movement, alternative contraction and relaxation of the muscular wall, or you can say that the gut wall layer, which, what is that, helps in, which pushes the food along the alimentary canal. So there we can say alternative contraction and relaxation, right, where it, what is it, the food will be there, it contracts and then it will going to move to the next part. As this will going to contract, right? This will be relaxed and that will keep on moving along the length of the alimentary canal. So that is the food which is there in the stomach is called as chyme. In the buccal cavity, it is called as bolus. Succus entericus, which is also called as intestinal juice produced by intestinal glands. The glands are called as the crypts of Lebercon. Right, chylomicron is that the finally absorbed fatty acids which will be absorbed into the lacteals in the form of chylomicron. Fatty acids absorb in the form of chylomicron. Protein coated fat globules, so which will be absorbed into the epithelium and then into the lacteal, that is lymphatic vessels of the villi. So match the column 1 and 2, right? And select the correct option, right? So what is that exactly? The lipase which acts on, right? Whenever we're going to say that the fats, which we can also say that lipids, right? So lipase, or I can say lipases acts on fats. And nucleus, we know that nucleus acts on nucleic acids. Nucleus acts on nucleic acid. Nucleus acts on nucleic acids and carboxypeptidase. We mentioned no that acts on proteins, peptones, proteases, and the dipeptide which acts on dipeptidase is an enzyme which acts on dipeptides. So answer for that A2 here we have, and in this option we have, and the B3 here also we have, and these two, what is it, the eliminated, and C1, sorry, C4, that is we have here but here it is not there. So answer for that is D is the answer. Here D is 1. We've checked that. So answer is D for this question. So let us continue in the next session. Right? Let us continue in the next session. Right? Thank you.